All right, well, welcome everybody to our Lipid Maps webinar. Thank you all for joining us. It's going to be presented today by Lisa Butler from University of Adelaide, but speaking to us from Chicago today. It's going to be identifying and targeting lipidomic drivers of aggressive prostate cancer. My name is Valerie O'Donnell. I'm based at Cardiff University in the UK. As usual, it's being recorded and it'll be online in the next 48 hours. We're going to have 45 minutes approximately. Lisa, up to you entirely, allowing maybe 10 minutes at the end for questions. And I've been asked by Lauren to tell everybody that you need to use the chat function. The Q&A has been disabled due to technical difficulties on certain browsers. So use chat and post your question during the talk, because that means then I can choose you in the order you've posted them, and then we can unmute you and you can directly ask your question. So remember, chat, not Q&A. We're still getting used to Teams these days. So now I'd like to introduce Lisa. So Professor Lisa Butler is Programme Lead and Group Leader at South Australian Immunogenomics Cancer Institute at the University of Adelaide. She has a PhD in cancer biology from University of Adelaide and was a postdoctoral trainee in preclinical drug development at Memorial Sloan Kettering Cancer Center in New York. Her current research focuses on androgen signaling and lipid metabolism in prostate cancer and on biomarker discovery coupled to drug development. And she's established translational research programs leveraging her preclinical models and involving primary clinical samples, prostate biobanking, and proof of concept clinical trials. So all the way through from mechanistic animal work right through to clinical trials. Recent work that she's published is focused on topics such as circulating lipid profiles and their resistance to androgen deprivation therapy, how peroxisomal beta oxidation regulates lipid metabolism, promoting resistance in prostate cancer, regulation of fatty acid metabolism supporting prostate cancer, and how extracellular vesicles can act as mediators in prostate cancer. So this all really interesting sort of mechanistic topics of clear, really high relevance to patient treatment. And so I really look forward to hearing about this work today. So thank you, Lisa, for presenting, and I'll hand over to you now.
Fantastic. Thank you so much for that introduction and for having me as part of this seminar series. So it's very exciting to present to this group, and today I'd really like to present a lot of unpublished data to really kind of let you know some of the directions that we're heading in using the lipidomic profiling that we've been doing of clinical specimens in prostate cancer, and how that's hopefully going to point us towards not only some useful biomarkers for this disease, but also some therapeutic targets that we might be able to exploit. Now, my group, let's just make sure, there we are, has been focused for the last 20 years or so actually on studying the clinical behaviour of prostate cancer. And I'm sure for many on the call today, as we are in Australia, experience prostate cancer as one of our biggest health burdens in the country. So it's the leading cause of male cancer diagnosis in Australia, and it's the second leading cause of cancer-related deaths. So my own group has been interested in many years in trying to understand really across the whole spectrum of this disease, from the time that it's diagnosed, where there's the potential to cure due to local therapies such as surgery or radiation interventions, or to just watch and wait in a regime called active surveillance. But unfortunately, the problem here is that we currently lack the diagnostic tools to tell us whether men are likely to progress or not when they're diagnosed with prostate cancer. And this unfortunately means that many men are overtreated, but paradoxically, some are not treated aggressively enough. Now, prostate cancer is dependent on male sex steroid hormones or androgens for its growth and survival. And so because of that, if men start to progress after localized therapies, the standard of care is androgen deprivation therapy, in the absence or presence of also chemotherapy. And this is quite successful in the majority of patients, so they tend to respond for varying periods of time, from weeks to years, sometimes decades. But unfortunately, we do see that there's an inevitable relapse that occurs, And then this then prompts further waves of different hormone targeting therapies, and also now novel targeting agents and radioligand therapy. Now, while those are prolonging life and have been a great advance over the last 10 years or so, the story remains that that it does remain a lethal disease at this point, and this is called castration-resistant prostate cancer. And there's a huge amount of effort in trying to develop new therapies for this later stage of disease. But this is, of course, very challenging. It's very heavily pre-treated, and trying to make an impact on survival at this later stage of disease is proving to be very, very difficult. So what we've been focusing on increasingly is this earlier stage of disease before the horse has bolted, and trying to prevent and delay that progression that occurs, and potentially intensifying therapy where needed, with the ultimate goal of reducing deaths from lethal prostate cancer. And so that's what I'll really be talking about today. Now, you might wonder where lipids came into the equation. I'm certainly not a lipid biochemist by training. It actually sort of came about in terms of thinking about how do we better understand and predict the clinical behaviour of cancers. And where we came upon lipids was really from an understanding that we hadn't got as far as we expected by studying the genome, the transcriptome, and even the proteome to a certain extent. We've got plenty of biomarkers, but very few have actually entered clinical management, and unfortunately, there certainly hasn't been influencing treatment to the extent that one might have predicted. So we became interested in studying metabolites, whether those are carbohydrates, lipids, amino acids, other metabolites, because we felt that they were the ultimate readout of tumour behaviour, because it does still integrate all of this important information from those other omes that I'd mentioned before. But it also incorporates extracellular and host influences such as energy status, nutrient availability, genetic damage, exposures, dietary lipids, and of particular interest to us in the prostate cancer field, hormonal regulation as well. Now, it's been known for a very long time that lipid metabolism is androgen regulated. So this is some of the first data that was presented by Johan Swinnen's group, who's well known to many of you. And my own particular background and expertise was in studying androgen signaling in prostate cancer, and that's really how we got into this area. So culturing prostate cancer cells, and this is an example here of a cell line called LNCAP with androgens, results in a huge upregulation of lipid synthesis, and that can be visualized here by an all red O stain, for example. They just become full of lipid. And from those early observations, and with the advent of more molecular biology techniques, we now understand through the contributions of many groups, including a few from our own as well, that many of the enzymes that are involved in uptake, synthesis, and metabolism of lipids, shown in red here, are either directly or indirectly androgen regulated enzymes. And so that led us to hypothesize that these androgenic hormones can drive changes in lipid metabolism in prostate tumor cells that does give them a growth advantage and is actually required for their survival. And on the flip side, that this exposes some novel vulnerabilities that we might be able to exploit for prostate cancer therapy in a way that doesn't involve whole-scale suppression of androgen levels in the body. So with that in mind, we undertook to do some profiling of clinical prostate tissues. Now, the reason we did this is that a lot of lipidomic work in cancer in the early days was undertaken on, rather artificial laboratory models such as cell lines, and so and even mouse models to a certain extent. And we sort of felt that we really had to take the bull by the horns and try and do this in a clinical setting, which was certainly posed a lot of challenges, but we brought together a really fantastic team to try and tackle this problem. And so this was our initial team shown up in the right here, where we brought together experts in lipid biochemistry, including Johan, who's been a really wonderful collaborator over the last 10 years, but also lipid profiling in diabetes and cardiovascular disease from the Baker, clinical management of prostate cancer, molecular imaging, endocrinology, and androgen signaling. So it was a really diverse and multidisciplinary group that sort of put our heads together. And we also were able to capitalise on the resources of the Australian Prostate Cancer Bioresource, which is a really fantastic biobank in Australia, where we're collecting specimens from men undergoing surgery from prostate cancer, and we collect their blood and their tumours. And you'll hear a bit later in the talk that we also were able to look at dynamic changes in the lipidome through looking at ex vivo culture of small pieces of clinical prostate tissues. Again, we were able to benefit from the expertise that we had available in our team for quantitative bulk mass spectrometry-based lipidomics, both at KU Leuven and also at the Baker, as I mentioned. This enabled us to quantify and profile the lipids that were present in these samples, but we also took the opportunity to further develop expertise that we had here in Australia around MALDI mass spectrometry imaging of lipids within the tissue, because being such a heterogeneous cancer, it was really important that we had that spatial information to add to the bulk analyses that we were undertaking. And we also had to develop a number of approaches to integrate that data together and gain a picture of the composition of lipids in those tumours. And we wanted to see whether those reflected clinical parameters of the cancers. Now, this has been published, so I'm not going to go through this in detail, but essentially when we profiled samples from patients, so these were matched normal or benign and malignant specimens. At the time, this was the number of lipids that we measured. We obviously measure many more now in the closer to the thousands. But essentially what we found was that, as expected, in the bulk analyses of the lipidome in these tissues, there was a huge amount of variability between patients. But we were able to identify a number of lipid species that were very consistently and significantly altered in that transition from benign to malignant disease in patients. And so this gave us some interesting biomarkers that we could validate not only in our own internal cohorts, but also with external studies that had either been published or that we had access to. If we took the lens of focus out a little bit, we found that there were some broader scale changes that were really evident. So, for example, as we expected, we saw enhanced lipid synthesis. So there was more lipids in the tumour versus the benign tissue from these patients, even when we normalised for cell number. We found that there was an enhanced proportion of mono and polyunsaturated fatty acids in the tumours compared with the benign tissues. And we also saw evidence of fatty acid chain length elongation. And this is that key initial paper here. Now, I'm going to just summarise a significant body of work over the last 10 years from our consortium, and some of those links are down the bottom here, just to give you a flavour for what we discovered. Now, you heard at the start in the introduction that we've also had a focus on the circulating lipidome, and actually this has progressed really well because it's much more readily translatable than some of the work that we do in the tissues. So this part of the program was led by Professor Lisa Horvath from the Chris O'Brien Lifehouse in Sydney, and she's been doing a lot of work with Peter Meikle's group at the Baker, looking at the circulating lipidome in various patient cohorts. And what we found was that, in fact, this was highly prognostic for overall survival and for disease-free free progression, my apologies, across the spectrum of disease, right from localized disease through to advanced castrate-resistant disease. And through collaborations with cohorts at the Mayo Clinic and others, we were able to show that the lipidome was actually closely linked to certain genomic aberrations in those cancers. So, for example, amplification of the androgen receptor gene, or loss of P53, or RB status. So this was quite exciting, and that's actually led to a patent and also the development of a NATA accredited. I think that it's the equivalent is CLIA compliant assay for measuring a 3-lipid signature in patient samples, and this is now being incorporated into a number of international clinical trials as well, to further validate its prognostic value. On the tumour side of things, we obviously lag behind a little bit, but it's giving us a lot of information at a discovery level. As I showed you in the previous slide, the tumour lipidome is quite distinct from benign tissue within a patient. And while we're much more limited when we look at localised prostate cancer because there aren't a huge number of genetic aberrations, we were able to show, for example, that the lipid profile does identify patients with the most common genomic aberration, which is a fusion of the TMPRSS2 and ERG genes, which occurs in about 50% of patients. We combined our efforts on the circulating and the tumour lipidome last year to publish with Mary Ellen Taplin's team in the US, to show that these lipidomic features are predictive of response to androgen receptor pathway inhibition, so using enzalutamide in both ex vivo culture but also in patients from a neoadjuvant clinical trial, and we were able to show to validate this, and we'll be continuing to do that in future clinical trials. So these foundational studies really gave us quite strong evidence that indeed, as we had predicted right at the start of this endeavour, that lipids are going to be giving us information about the aggressiveness and the behaviour of the disease. But the reality was that these bulk analyses, don't really tell us about the source of those lipids, and that really kind of limits the mechanistic insights that we can glean from this, and also limits our ability to target them, because if you don't know the source, you don't really understand the biology behind those changes and where they're occurring. So this really led us down a path, certainly in my own group at SAMRI, where we've been doing a lot more spatial profiling of lipids as part of our discovery efforts. And this has been a fantastic collaboration with the mass spectrometry team there, with Martin Snell and Paul Trim, who would be known to many of you, who had a long-standing interest and expertise in spatial lipid profiling. And I think I always use this image here as a nice example of how important this is when you look at the complexity and the multifocality of clinical prostate tumours. So in the middle here, we have a tissue that has been annotated by a pathologist, and we've just sort of colour shaded the regions to make it easier to see. But essentially, these sorts of tumours comprise stromal areas here, sort of shown in the purple and blue. We have epithelial regions, and the benign regions are shown here in yellow, and then we have tumour regions that are shown here in red. And this is pretty typical of the types of samples that we get in our localised tissue cohort. It's always a mix. There's very few cases where it's absolutely chock full of tumour. So when we then go and do mass spectrometry imaging, which is essentially, I'm sure, is well known to this audience, but essentially a laser being fired in a grid pattern across this tissue and integrating all the spectra from each of those pixels into a spatial map. And what we show on the left here is segmentation of the data, essentially like a heat map, but in a spatial setting, where these colours here represent areas that have very similar lipid composition. And you can see here very clearly that when you do cluster according to lipid similarity, it aligns very, very closely to the morphological features of the tissue, with the tumour here in red, that benign epithelium in yellow, and the stroma in blue. And you can go even further than this to look at parts of the immune infiltrate in the cancers, or other specific pathological features such as cribriform forms of cancer. So we wanted to use this approach to see whether lipid composition in the prostate tumours, or the surrounding tissue microenvironment, which is something that's largely neglected in these sorts of studies, would that tell us something about how aggressive that disease is going to behave? So to do this, we were able to make further use of the bioresource that I mentioned to you earlier. And so in this case, we weren't just interested in looking at particular pathologies or benign versus malignant disease. Here we wanted to capitalise on the fact that we've invested a huge amount of time and effort over the last 10 to 15 years in obtaining detailed clinical follow-up on the patients in our cohort. So this is a bit of a summary on the left-hand side here, where we have almost 5,000 patients in our biobank, of which 1,300 have frozen tissue, which is obviously the only type that's going to be suitable for lipidomic analysis. From the clinical follow-up data, we know that the vast majority were, we say this word carefully, but were cured in the sense that they've experienced no recurrence with at least 8 to 10 years follow-up. But then we have some that have experienced that PSA relapse, so that relapse in terms of seeing an increase in the blood marker of prostate cancer called PSA. Now, a number of patients that experience this PSA relapse will never really go on to have clinical progression. And so they're sort of in this group here on the right. But what we were most interested in is these patients here that did experience what we call a clinical or metastatic relapse. And so that means that they had documented evidence of metastases by imaging and or had died from prostate cancer, because these clinical relapse patients have been shown to be an effective surrogate for overall survival in prostate cancer. So we basically set out to undertake a comparative analysis between these non-relapsed and relapsed, clinically relapsed groups of patients. And today I'm going to tell you a little bit about the pilot study that we had done with a cohort of 47 of these men. So again, just to remind you that we have the samples that are from the surgery of these patients. We then follow them for at least 8 to 10 years in the case of the no relapse patients, and classify them accordingly. We then did a fairly detailed sectioning plan so that we could undertake spatial profiling, but also bulk lipid profiling from the same samples, because, as you're all aware, some of the ambiguities around the identification of samples from mass spec imaging. So spatial imaging we undertook on the timsTOF Flex, and did bulk analysis with Johan's team in Leuven. I just thought I'd mention as well that as part of this, like many biobanks, many of our samples that were frozen in the OCT embedding media. And this is obviously causes a lot of havoc when you're doing mass spectrometry. But, you know, between when we started this project and now, we now have some pretty good techniques to kind of account for this. So our PhD student, Jacob Trung, developed and published a method for washing this OCT medium from the slides without impacting terribly much the lipid profile that we obtained. And so this has not really opened up not only for our project, but I think biobanks worldwide that largely have OCT embedded tissues. Now, again, we probably don't need to explain to this group the reason why this bulk versus spatial approach is so important, because we are really needing to understand that information about the tissue architecture, where the source of those lipids are, and kind of understanding that what we see in a bulk analysis is extremely susceptible to the features of the tissue, how much epithelium is in that particular tissue and how much of that is tumour. So using that smoothie analogy, it's very difficult difficult from this part of the smoothie to really understand what the component fruits were, what their quality was, and how they related to each other. And it probably then wasn't too surprising, but was extremely disappointing to us that from the bulk lipidomics analysis that we undertook, which, as you know, was quantitative and very well annotated, unfortunately we saw very little in the way of a relationship between the lipid composition of those samples and the outcomes. So red and blue here are clinical and non-relapse respectively, and you can see there's really no relationship there at all. However, it was a very different story when we went to the spatial analyses. So just again highlighting that the work that I'm presenting here is from Jacob Trung's PhD project. And here's an example again of the types of analyses that we undertook with these tissues. I showed you earlier that after doing the annotation of these tissues and comparing that to the clustering of lipids from the mass spec imaging data, when you cluster into two key areas of similarity, it splits immediately into stromal versus epithelial areas of the tissue. If you take that cluster further into three, you start to see the benign pulling away from the tumor. So those are always the three strongest drivers of difference in lipid composition. And this then enabled us to identify and compare lipids that were primarily localized in tumors, for example, versus benign disease, but also in the stromal areas, which is, as I mentioned, something that's often really neglected in biomarker and other similar studies. So, in fact, here we did see a very clear difference in the tissues from patients who had relapsed. And so, for clarity, I'm just going to present today the results from the tumor and the stromal tissue from these patients. So if we look here in the red on the right here, this is the tumors from patients who had experienced clinical relapse. And you can see that they're really quite metabolically distinct from all of the other samples, but particularly from the non-relapse tumors here. Interestingly, we did see changes in the stroma as well, and so that's something that we're actively following up. So if you look at this with a fairly typical plot here, you can sort of see that we can compare and identify lipids that are enriched in tumours that had progressed versus those that have not. And we can also, these are ones that I'll talk about a little bit further in the next slide. But we can also do this in the stroma. And I think this is an area that we really want to push forward with. And certainly we collaborate very closely with Massimo Loda's team at Weill Cornell in New York, who has shown huge differences in the metabolism of stroma and how important that is for the aggressiveness of cancer. So this is something we're keen to keep working on. Now, a lot of these data are things that we're working through, but what I thought I would highlight today is, as a bit of a sanity check actually, is that we saw some observations that really made sense to us biologically from studying lipid metabolism in cancer. So we found that in the tumours from patients who had experienced a clinical relapse, we found a preponderance of lipids that contained the same fatty acid, so 16-1 fatty acid, palmitoleic acid. And so this is an example here of the actual imaging. So a patient up the top that had not relapsed versus a patient at the bottom that had. And an example of 4 different lipids here, all containing 16-1, that were preferentially abundant in this clinically relapsed patient, but were really not evident hardly at all in patients that had experienced no relapse. And of course, this is just a couple of illustrations here, but if we quantify this over our cohort, we found that this was actually incredibly consistent. And as I mentioned, consistency is not something we normally see in tissues. They're normally extremely variable. So, when things like this pop out, we really take notice. So this shown here, the red is our clinically relapsed tumours, non-relapsed tumours in the blue, and here's our stroma on the right. Now you see in the stroma here, we're not really seeing any enrichment, certainly with relation to outcome, and in fact the abundance was very low in the stroma anyway. But in these patients across the board, these were elevated, and in fact all of these lipids containing the 16-1 were highly correlated with each other, both in terms of abundance but also spatial localization. Now, this was also seen when we then went back and looked at some of the lipids that we'd identified either in the tumour epithelium or the stroma, and we were able to show that indeed these were associated with clinical progression, both with using a clinical relapse endpoint or that earlier biochemical relapse endpoint. And working with our biostatisticians, we've been able to show that when we adjust for batch and Kappa-S variables, which are some of the clinico-pathological variables in these patients, these provide additional information beyond those known prognostic factors. So our focus now as biologists really is to try and see that whether these apparent lipid biomarkers are purely giving biomarker value, or whether they are in fact revealing a biology that's important for poor prognostic disease. So can we link those back to the drivers of these lipid profiles? So again, using the example that I just showed you of the palmitoleic acid, this is obviously produced by the activity of the enzyme SCD1. And so work from another PhD student in my lab, Julia Scott, who's now in the Netherlands, has shown that in fact this is a potentially useful target in prostate cancer. So she was able to look at, this is where we do go back to cell lines to look at mechanism of both the LNCAP and also a treatment-resistant subvariant line of LNCAP, MR49F. And she treated those with an SCD inhibitor, one shown here, but she's also confirmed this with an independent inhibitor and also by knockdown genetically of SCD1 as well. And she was able to show that, in fact, it was quite effective at reducing the proliferation of both of these cell lines and inducing cell death, shown down the bottom here. But in a non-malignant line, PNT1A, this actually had very little impact on proliferation or death. So that wasn't particularly surprising. SCD1 has been shown as an important target in prostate cancer previously, but it gave us a lot more confidence that we're identifying pathways that had biological significance. Importantly, you'll hear a little bit more again about this. We do a lot of work in tissues, particularly to translate our cell line work into a more clinically relevant context. So from fresh specimens, we're able to culture those as explants in the absence and presence of an SCD inhibitor. And again, Julia was able to show that the proliferation of these tissues was significantly inhibited by this agent. And there was also an induction of cell death in these tissues. So this gives us some confidence to go forward and try and identify some of the other drivers, particularly of some of the really novel lipid classes that we identified that haven't been linked to either cancer or cancer outcomes before. One of the ways that we're proposing to do this, obviously spatial analyses are very expensive normally, and particularly when it comes down to transcriptomics or epigenomics. And so a way that we've been working on this a little bit has been to harness the abilities of laser capture microdissection, and the fact that we can do this on the same sections that we do our lipidomics on by sectioning our tissues onto pen membrane slides. And this is again some work that's been published by Jacob last year. So based on lipid profile, he was able to undertake this very detailed microdissection of tissue and was able to achieve thousands of proteins being detected and identified in particular regions of these tissues that he could then relate to to the lipid profile in that same tissue. So, for example, if we looked at the abundance of an enzyme here, LPCAP1, we can also then look at its inferred activity as the ratio of abundance of its product to its substrate. And so see that that lines up extremely well here, particularly in the cancerous epithelium of the tissue and not at all in the benign epithelium, confusingly here in blue. We can also quantify this. So this is an approach that we want to be able to use on a much larger scale in this cohort and also independent cohorts to both validate the findings and the associations we've seen, but also to do some discovery in terms of the driving pathways. So in summary, I hope I've shown you that the spatial imaging of lipids has enabled us to find some very useful characteristics of the various components of the tumour microenvironment, and which of those are potentially linked to the aggressive features of those cancers, with the opportunity that if we can identify tumours that are likely to be aggressive early and link those to targetable alterations with a metabolic focused agent, we might be able to prevent or delay that progression to that much more advanced incurable stage of disease. And I think it's important to know that it's not just in these known high-risk patients, as the analyses that we did with our biostatistician showed. We hope to be able to also identify men that are likely to have aggressive disease that, you know, currently would be viewed as having very low or only intermediate risk of progression. So for the remainder of the talk, I'd just like to bring in, there's some points here for discussion that we can maybe come back to at the end. Particularly in this audience, I'd be keen on thoughts around really adequately capturing the complexity of a big tissue with, you know, single slices or small regions that are used for undertaking the bulk analyses. But also approaches to data analysis remain a real issue. And because we are able to do much larger batches with mass spec imaging, batch correction has remained a really important issue. So some of the other work that we've been doing that you might be interested in has been looking at those dynamic changes in lipid composition in tissues. And we're particularly interested in, from my own background, in understanding how androgens affect the different types of tissue, cell types within the tumour microenvironment. So to do this, we utilise the ex vivo culture model that I showed you earlier, so explants of fresh tissue, which we obtain from surgery. They are collected on ice and taken back to the laboratory, where we cut them into small pieces and culture them in a way that maintains the structure and the hormone responsiveness of the tissue as it was in the body. So essentially the tissues are placed on these small gelatin sponges shown on the bottom left here. And the reason for that is to give it a scaffold. If you don't put these tissues on a scaffold, they end up being covered and encapsulated in fibroblasts, and they also lose a lot of their steroid hormone receptors, which is exactly what we don't want to have happen. So the good thing is we can add in various treatments into these wells in a 24 well plate, so you can have multiple samples per patient. And then you can look at different time points, different treatments, and do an within patient comparative analysis. And that again cuts out a lot of the noise associated with the study of clinical tissue. We can also collect the culture medium as well to look for things like secretion of PSA and other factors from the tissue. This is just a small figure here just to show that this method of culturing tissue retains both the expression of the androgen receptor and also its activity as measured by its direct target gene PSA. Now, another thing that we've been doing a little bit more with the mass spec imaging has been to localize and visualize the drug penetration into the tissues. And this was something that we were asked about for years. And we always inferred that it was penetrating fully because we were seeing when drugs were efficacious, we tended to see its effect on proliferation, for example, across the whole tissue. We didn't see areas of difference in response. So, but working with Martin and Paul really gave us the opportunity to start to directly visualize and look at the kinetics of penetration of drugs into our tissues. And we could also look at metabolism, because we could look at some of the known metabolites of these drugs. So this is an example of how we look at androgen signaling in a tissue. We use in this case an agent called enzalutamide, which is an androgen receptor inhibitor. So it's an antagonist of the receptor. And so what you can see is that, in fact, it suffuses the tissue fully, but it doesn't just appear everywhere. It's very strongly localized to the epithelial regions of the tissue, which is where its intracellular target, the androgen receptor, is most strongly localized. We know that we're hitting the right target in these tissues because when we undertake bulk transcriptomic analysis, we find that the top pathway reduced is androgen response. And unsurprisingly, we see a huge amount of modulation of metabolic pathways within the tissues. And interestingly, because these tissues do retain the immune infiltrate from those patients, we do see often enhanced inflammatory responses as well, which is something that we really should be focusing in on a little bit more. So just like I showed you for the static tissues from our biobank, targeting androgen signaling through this ex vivo culture approach did enable us to look at specific changes that were associated with either, an inhibition of AR or a knockdown of AR in the case of an androgen receptor degrader. And again, we can identify potential lipid species that appear to be selectively abundant in either the treated or the control samples. And we can compare on the right-hand side of the slide here between these and relate that to, again, the various morphological features of the tissue to identify in tumour cells or in the stroma particular lipids that are consistently altered across our patients. And so again, that gives us some interesting biomarkers to study, and in some cases those may be detectable in the blood, which would certainly solve a lot of the sampling issues. But again, as we sort of did earlier with our tumour lipidome work, we sort of wanted to move away from overly focusing on individual lipids, because I think, that a lot of these are interrelated. We don't want to over-interpret a single particular lipid as being particularly prognostic, particularly if it's reflecting a broader biology. So work from Maggie Centenera, senior postdoc in my lab, was able to show that, in fact, one of the most eye-catching things that we saw when we compared his 19 patients here, plus or minus that inhibitor that I showed you before, enzalutamide, and when we did the heat map of various lipid classes here, we're just showing the PC class here, ordered by saturation group as well as by chain length. This sort of striation pattern that we saw throughout the heat map across multiple different lipid classes was indicative of a shortening of the fatty acid chains in the lipids, in the various phospholipids from these tissues. So we're seeing, kept seeing the same effect of a shortening, a reduction in the long chain fatty acids and a relative enhancement of the short chain fatty acids. And so this was something we felt we couldn't ignore. So we didn't focus in on individual lipids. We just focused in on this overall phenomenon of lengthening. Now, the other reason that we got excited about this was when we went back and compared to our initial results with the lipidome from the clinical samples. Again, one of the most notable features that we saw comparing malignant with benign tissues from the same patients was this elongation of fatty acid chains. We hardly ever saw shortening. It was always elongation of fatty acid chains in the tumour samples. And then, of course, when we looked across the literature, and certainly Johan's group has done a lot of work in other cancers as well, it really is a feature of a number of different cancers, such as lung, melanoma, and breast. And then we and others also showed that it tended to be a feature when prostate cancer cells in laboratory models became resistant to targeted therapy. disease. So it was sort of something we couldn't ignore, but it was sort of surprising because it's obviously a very carbon and energy intensive process for the cell to undertake. So there must be some sort of advantage to the tumour cell in doing this. So again, sorry for this audience, this is probably very basic, but this elongation, just to remind you, is essentially is talking about the sequential addition of 2 carbons to fatty acid chains, and it's catalyzed by a family of enzymes called elovals, and there are 7 elovals in mammalian cells. And I won't talk about this in detail because this has been published, but Maggie's work showed that in prostate cancer, at least, eloval 5 was the key elongase that was not only required for the androgen-induced fatty acid elongation in prostate cells, it was also really important for the growth and survival of those cells as well. So it seemed to be really important for promoting tumour progression. And indeed, when we went on to stably overexpress eloval 5 in LNCAP cells, so essentially mimicking advanced, more aggressive cancers, we did find that they grew faster, they were more proficient at forming colonies, and were able to invade much more effectively. She was also able to show, again, in our tissue context, because we always want to take our discoveries from the tissues back to the tissues when we're targeting. If we tried to block this elongation either directly through nanoparticle-delivered siRNA against eloval 5, or pharmacologically shutting down this entire pathway by inhibition of ACC1, she was able to show that that this was very effective in the tissues at blocking proliferation. So I'm just showing the results here for the eloval 5 knockdown. So you can see we had a pretty reasonable knockdown of eloval 5 expression by just over 50%, but we really effectively blocked proliferation in those tissues. So this has now led to an ongoing joint program with Johan's group to develop some selective eloval 5 inhibitors, because we feel like they're potentially going to be quite useful in a prostate cancer and potentially other cancer setting. So for the remainder of the talk, I wanted to just, I guess, describe to you some of the work that we've been doing, trying to understand the mechanism by which eloval 5 is promoting growth of and elongation in general, really, of prostate cancer. And this has been a long-term collaboration both with Johan and with Massimo, which has been funded by a number of grants, and we also have exchange programs of master's students between our labs, and that's been really beneficial in moving these projects forward. So just to let you know, so each eloval has its own specificities, and so for eloval 5, it's particularly involved in elongation and synthesis of MUFAs and PUFAs. So its MUFAs product is one called cisphexenic acid, and the PUFA products mainly that are studied in the context of eloval 5 are arachidonic acid and DHA. And I've just summarised some work here from Julia, showing that in fact these products and substrates of eloval 5, particularly the monounsaturated ones, are particularly important for survival of prostate cancer. And I've just shown and linked to some of the work here if you're interested in looking at that a little bit further. But I just finish by showing you the work that we've been doing to sort of use a hypothesis-driven approach to look at a potential mechanism, which is through the elongation of lipids being able to influence the membranes, not only of the plasma membrane of the cell, but also that of the organelles. And so we focus very much on the endoplasmic reticulum because elongation of fatty acids in the phospholipids in those membranes would be anticipated to generate thicker and more disordered membranes. And we know from the yeast field that this can actually activate an unfolded protein response, which might be contributing to the survival level of the tumor cells. Unfolded protein response really, again, doesn't need much introduction here. We know that it's a series of 3 parallel pathways that ultimately promote cancer cell survival, largely studied in the context of proteotoxic stress, but we're interested here in studying lipid bilayer stress and as a potential activator of UPR. So our student Deanna Miller was able to undertake a comparative analysis of cells that were overexpressing a level 5 versus those that were not. She looked at whole cell lipidomics, but also ER-enriched lipidomics, and matched that together with a transcriptomic analysis. Now, as expected, what she found was that when we looked at the whole cell, that overexpression of a level 5 caused a relative increase in those longer chain fatty acids in the cells. So we were seeing an elongation phenotype as we expected. When we looked at the transcriptomic data, we were able to see that indeed unfolded protein response was one of the key pathways enriched by a level 5 overexpression. And many of the known genes associated with each of the 3 arms of the UPR pathway were enhanced in the level 5 overexpressing cells. And we also managed to confirm this at the protein level. We were able to also show that these changes did indeed affect the structure of the ER. We started to see these ER whorls, which are known to be associated with ER stress. So we saw this in our overexpressing cells, but we're also able to see it in advanced clinical tumours as well, to give it some clinical relevance. We were able to also do selective enrichment for the ER using a specific kit that we were able to then do the lipidomics on those samples, and again just confirmed that the lipids within the ER membranes were indeed showing evidence of elongation as well. Finally, through some collaboration with Guillaume Thibault in Singapore, we were able to use his yeast models, which had been selectively engineered not to be able to respond to proteotoxic stress, to show that we were indeed, with the yeast homologue of a level 5, we were able to induce a UPR purely by overexpressing that level. So it seemed that there was a plausible basis for this hypothesis. Overexpression of a level 5 therefore rendered the cells relatively resistant to ER stress. And this, the enhanced proliferation that we could see with a level 5 overexpression was completely blocked by inhibition of each of these arms of the pathway. Conversely, I know we're running out of time, a level 5 downregulation was also able to reduce the expression of these various proteins involved in UPR. Most notably, ATF4 was the most consistent change we saw across the entire pathway. Set of pathways, sorry, and indeed we found that knocking down a level 5 also sensitized to ER stress induced by tunicamycin, and we confirmed this in another set of cell lines. Finally, we wanted to relate this back to androgen signaling. So androgen signaling has been known to influence directly the UPR by transcriptional upregulation of genes involved in the UPR, and we indeed confirmed this ourselves in a couple of cell lines. We also then found that the increase in proliferation in prostate cells that has been well known to be caused by androgens could be blocked if we either inhibited the entire 3 UPR pathways or each one individually. So UPR is very important for androgen action in prostate cells, was what we took from this. Now, fascinatingly, when we looked at the influence of a level 5 in this process, we found that, and I've just put some red boxes here to help talk you through this Western blot, knocking down a level 5, as we had shown before in 2 different cell lines, was able to reduce the levels of these UPR proteins in the prostate cancer cells. When we cultured those cells with androgens, we saw that known induction of UPR. But most fascinatingly to me and surprisingly was just by knocking down a level 5, we were able to completely block that induction of UPR by androgens. And so this was quite an unexpected finding with only one enzyme being able to influence this so strongly. So what's the functional significance of this? Well, as I showed you, androgens induce proliferation of prostate cancer cell lines, but we were able to block that with a level 5 knockdown, which was really, really exciting, as well as by pharmacological inhibition of the level 5 pathway. So what we took from this finding was that, in fact, androgens are often thought of as, binding to their receptor, transcriptionally activating a number of genes that are required for the growth and survival of prostate cells. But I think what we're showing here is that, this key elongase enzyme that is induced by androgens in prostate cells is actually influencing cell membranes to be a really important part or aspect of the survival and proliferative benefits induced by androgens. So this is really encouraging us to think in a slightly different way about how androgens act in prostate cancer cells through their effects on cell membranes. And so I think it gives impetus to further developing and refining the methodologies that we have to look at cell membranes and how they're influenced in cancer cells. So, and something that will, I think, continue to improve as technology does. So I'll leave it there. So this is just to acknowledge my lab as well as our funding bodies. I've mentioned most of the people in the lab that did the work so far, and these are the key collaborators and, of course, the clinical resources without which we would not be able to do this sort of work. So thank you again, and sorry I went a little bit over time, but hopefully we've got time for a few questions.
Yeah, great. Thank you. That was really interesting. I mean, I really love the way that you used lipidomics initially to, you were looking for biomarkers for describing, who's going to, do better with disease or not. And then it moved into using that information to discovering mechanistic insights, which is really, really nice. So I have several questions, but I think the first one, Kevin has a question.
Absolutely amazing talk. Very cool and interesting research. Really amazing. So I had a small question from the beginning of your talk where you said that in the tumor, it also has, in general, more lipid production based on the different subclasses and classes of lipids that are there. Can you also say something about where it is funneled into? Is this just mitosis that is extra, more cell growth itself, or is something extra going on, like an adaptation to immunity or hypoxia or even enlargement of lipid droplets?
Yeah, so that's a good question. So no, we haven't done a huge amount on that yet. Something that I didn't mention in the talk that we are trying to look at is to do more isotope tracing in our tissue studies, because I think that will tell us a little bit more about how these tumors are both synthesizing, taking up, and utilizing lipids. So that's something that we've been developing. We've done it pretty well with glucose and glutamine. With lipids, that's taking a little longer to optimize, but that's where we hope to get, so that we can be kind of really dynamic in what we're looking at. Because I think the trouble is, like anything, these are a snapshot, so they're not telling us. They're telling us something, but they're not telling us the whole picture.
But in the snapshot, you could already say that certain big groups maybe change. For example, the triacylglycerols or cholesterol or whatever. Are these very much affected, or is it more the PC, sphingomyelins, more the...
Yeah, sorry. So yes, we do see definitely increases in membrane lipids. So that's, I guess, pointing to effects on proliferation. What we haven't done as much as we really should have is characterize the lipid droplets in our samples. I mean, a lot of other groups have done that sort of thing, but we should probably do that and relate it to the lipidomic profiles that we get. Yeah.
Okay, great.
Very nice research.
Great.
Thank you.
So yeah, I've got several. So going back to the earlier part where you talked about 16.1 and SCD1, it was interesting that 16.1 seems to be changing across multiple lipid categories, which, normally I look at this kind of data and I'm thinking, right, all the PCs should be doing one thing, or all the PEs, but you're looking at one fatty acid free and in multiple compartments seem to be you know, and that was quite intriguing. It was like specific to one fatty acid. Why? What's special about 16.1 that?
Yeah, that's the big question, and it was again one of those things that it just really hit us because it was very clear from the data that we generated that this was something very consistent. And when we did those correlation analyses, they were very, very tightly linked together. And I think that's an example of what I was talking about earlier with not being overly invested in specific lipids as biomarkers, because a lot of them are reflecting something that's going on across multiple lipid classes. So, and that certainly seemed to be the case here. So, and, you know, I think there's some interesting opportunities with that 16.1 because it is detected in the serum and plasma as well. So, you know, it is something we could potentially follow up, but obviously it's not as novel perhaps because it has already been linked to cancer, but it does suggest that it's possibly, you know, the activity of this enzyme that's then ending up in a number of different lipids throughout the cell.
Maybe, I mean, but that enzyme will also metabolize 18.0 to 18.1, and you didn't see the same with that, did you? It was just 16.1.
No.
So yeah, I mean, that is quite intriguing. Is there something specific that 16.1 does in a signaling mechanism that we don't know? Or so that was one. Then another was about the higher levels of PUFA that you saw, because maybe it's quite unrelated to this, but there is literature, isn't there, and quite a bit coming out about the N6, N3 balance and how that might impact on cancer, mainly kind of dietary intakes and things. So if you're seeing higher levels of PUFA because of VLOVL5, that's going to impact both N3 and N6. I mean, is there changes in the balance of those?
Not that we've seen, no. So, so it does seem to be in general because, it influences both. But we think in terms of the effect on cell viability, we were sort of surprised actually because LIPID-5 in particular is mostly studied in the context of PUFAs, but it was actually its MUFA product that was more important. So we couldn't rescue our phenotype with AA and DHA when we're blocking a lot. Level 5, but we could with cisphexanic acid. And so we've sort of got a story building on that to show that in fact this cisphexanic acid is very important. It's not just important because it's a monounsaturated, because there's obviously been a lot of literature now about monounsaturated fatty acids protecting against ferroptosis and the like. So we couldn't rescue, for example, with its structural isomer oleate, but we could with cisphexanic acid. So this particular product of a level 5 is really important. So, but yes, the PUFA levels are one of those sort of interesting things that, you know, it's definitely some sort of advantage to the cell, possibly in terms of membrane fluidity and the like, but it obviously brings that danger of lipid peroxidation and susceptibility to ferroptosis.
And that brings me on to this, you know, there's a lot of old work from Ken Hahn about the 12-lipoxygenase in prostate cancer and 12-heat playing a role. And of course, some of these will be substrates for not just 12-heat, but other similar lipids from DHA, EPA, whatever. So have you looked at that? Could that be part of anything to do with this story of the long chain PUFAs and the advantage they're conferring 12-heat? Have you?
Yes. Almost certainly. We did some early kind of exploratory studies, and we did see some effects on 12-heat, but we haven't really followed it up, to be honest. So I think potentially it is going to be really important. We've sort of focused on these fatty acids at the moment, but yes, I think you're absolutely right. I think that's going to be very important.
Yeah, their metabolites. Yeah, maybe knocking down ALOX12 or whatever. So. There was another question. About two other things. One is, I guess, around clinical translation. So it's great to hear that some of these lipids might be then, you know, used for biomarkers for patients. But the challenge of having, because, you know, if it's going to be translated clinically, it's not looking at fold change anymore, is it? I mean, it's really highly validated quantitative assays, and that is a challenge for these kinds of lipids, isn't it? And how is that? Progressing.
Exactly.
Yeah.
So in terms of the tissue side of things, I think that's always going to be quite a challenge because, you know, it's so dependent on sampling. It's so dependent on platform. You know,
reference values, all that. Yeah.
Absolutely, it's going to be very challenging. And so where we've I think made some strides has been in the circulating side of things, because certainly our collaborators at the Baker have some really well-validated platforms that have been used for quite a long time in a non-cancer context. And of course, even in inborn errors of metabolism, there are assays, established for things like acylcarnitines and the like, so which are other prognostic classes that we have identified as well. So the ones that I mentioned, they managed to sort of really refine down their list of key prognostic lipids to just 3, which essentially captured most of the prognostic value of the larger lipid signature. And so they were able to develop this assay that is now clinically validated, that theoretically should be a much more, consistent and useful assay clinically. So that's why it's now being sort of analysed both in retrospective cohorts, but also in prospective clinical trials coming up, just to see how well it performs and whether it continues to identify groups of patients that would benefit from an additional intervention. So, for example, one of the key features of these prognostic lipids in the blood is the abundance of ceramides and sphingolipids, and so that finding has now led to a clinical trial where patients with a so-called poor prognostic lipid signature, so elevated levels of these ceramides and sphingolipids, will then be randomized to receive a sphingolipid targeting agent, for example. So that's sort of now leading to design of trials to specifically test whether these lipids are purely of prognostic value, or predictive value, which is still a huge value in and of itself, something we don't have. But if it's also flagging a biology that's targetable, then that makes it kind of even more exciting, because then it's got a biomarker linked to a potential therapy. Yeah,
great. Okay, and one last thing before we go, because I know we are over time, but it's just been really interesting, everything that you presented. Is around, and I know it's related but not related. I mean, this is the iKnife work that was talked about a number of years ago, and the use of mass spectrometry as a way to surgically distinguish tumours and during operations. So has that progressed into clinical use? Do you know? Or
look,
I've lost track of that.
Haven't. No, I know it was something that was talked about a lot, but not in. I certainly haven't been hearing about it in recent years. So maybe others on the call might have heard more, but no, not yet.
Yeah. Okay. Well, I think that's it. We're out of questions, and thank you very much, Lisa. That was a fascinating talk, and best of luck with your work, and thank you for everybody for attending today, and we look forward to seeing you on the next webinar. Bye now.
Right. Bye-bye.